morning children and welcome to the online teaching session. Students, today we'll be starting with a new topic named as processes of recombinant DNA technology. Okay, now see what points we have already discussed in this. In case of recombinant DNA technology that we are already studying from the previous lectures, what to do? At first, we will isolate the DNA. So your first topic will be isolation of DNA. Now how the DNA will be isolated? It will be isolated by some enzymes. And after that, there will be fragmentation of DNA. Again, with the help of some restriction endonucleases, we will cut the DNA into pieces. Then your third step will be isolation of fragmented piece or desired DNA. Okay. Then next step, ligation of DNA into the vector. Okay. After that, your next topic will be transferring of desired DNA into the host body. Clear? And then culture it. Okay, and after that we will do some next other steps, and that will be extraction of desired products. Okay children, now see these points. The first point is isolation of DNA, and this we have already done with the help of using some enzymes according to the plant, animal, fungal cell and bacterial cell. Clear? After that, cutting off the DNA into the specific site, at least 2 sites, because we have to take out the piece of the DNA, and that will be done with the help of the molecular scissor. And we have already studied about the molecular scissor and the types of molecular scissor. After that, we have studied that how the molecular scissor or restricted endonuclease cut at the site, and we have seen that it cut either at the blunt site, coarse site, sticky site. This I have explained to you. After that, ligation of the DNA into the vector. We have to insert the DNA into the vector. Clear? Then, transferring of desired DNA into the host cell. After entering the DNA into the vector, we will transfer it into the host, and the host will multiply. Then our desired DNA will also multiply. We'll keep the host in a specific medium so that it can get all the possible environment and conditions for the development. So if the host will multiply, then our required DNA will also be multiplied at a large scale. Then our job is to take out everything from the body of the host and just to package it and sell it into the market, just to make the desired product so that we can sell. So this whole is the process of recombinant DNA technology. Okay, now children, the point is we can do isolation, fragmentation of DNA, then isolation of desired DNA. Clear? Now, before the isolation of desired DNA, we need the multiple copies of the DNA. Okay, now suppose from here we can see some points like this point is okay, this point is okay. Isolation of the desired DNA into the, okay, desired DNA has been done. Ligation of DNA into the vector. Before going this, we need the multiple copies of the DNA, children. Why we need the multiple copies of the DNA? Suppose if we talk about any crime scene, clear? And any crime has occurred and these detectives and they all have got a small piece of blood sample. Now, they want to detect the culprit, the criminal. And just to identify that, they will take the blood sample and go for the DNA testing. Just to find out the sequence and find out the criminal. That is the job of all the CBIs and detectives. Okay. Now, if they will take out the blood sample, then the blood contains 3 types of cells. RBC, WBC and platelets. Out of this, RBC and platelets, they do not contain any nucleus. But WBC contains the nucleus, so it will have the desired DNA. Okay, but we all know that the number of WBCs are very small in comparison to the RBC. So already it is a drop of blood and the number of WBC is also very less. So how to detect, how to find out the DNA? Now suppose if they will test for the DNA and in case the result goes wrong, then what will happen? They will waste the whole sample. Okay, so what to do? They need to store the multiple copies of that DNA so that if the test fails, then also they all will be able to get more and more sample to test. Clear? Now see, just to make the multiple copies, that is before we begin from this step, ligation of the DNA into the vector, what they will do, they will make the multiple copy of DNA. And this multiple copies can be done with the help of a technique named as PCR. Okay, and the full form of PCR is polymerase chain reaction. Now today we will begin with the topic polymerase chain reaction so that we can go for the next steps. Clear? So let's start the new topic, that is polymerase chain reaction. Got my concept, children, that what I have to, how to begin with the PCR. Okay, we need the multiple copies of the DNA. Clear? So, now the topic is PCR. Okay, I'm writing here. This is polymerase chain reaction. So our today's topic is polymerase chain reaction. In the first step, what we do denaturation. The first step of PCR is denaturation. Okay, now see children, what we can do in the denaturation. In case of denaturation, we have to break our DNA into its 2 fragments. If you remember that, you have already studied about the DNA replication. That is how the DNA opens up from one end and the new piece of the DNA gets formed. Okay, I am telling you about this Okazaki fragment. Okay, I have already told you about this, that there is a formation, then we will have the nucleotides, okay, and it will form the two pieces of DNA and we will get the copy of the DNA. This is only the PCR, okay. As it happens in our body by natural mechanism, we can do the same, or our scientists, they have developed the design to do the same in the laboratory also. They have increases the number of this DNA so that they can keep the safe copy. Now, what to do? The first step is denaturation. And the meaning of denaturation is just to cut the DNA into fragments. Now, suppose children, this is our piece of DNA. And if this is DNA, this is 3rd prime, 5 prime, then 3rd prime, 5 prime. We all know that it is anti-parallel. Okay. Now what to do? We have to cut this hydrogen bond because they all are linked with the hydrogen bond. This also we know very well. So if I am drawing it, you can understand. Yes, that nitrogen bases. If you remember, okay, I have already taught you these things. And second one is in the opposite direction. Okay, it should be little bit up like this, and they all are linked with one another. So if this is 5 prime, then this will be 3rd prime. Clear? If this is 5 prime, this will be 3rd prime. 5 prime, 3rd prime. Now, okay. Now they are anti-parallel. The same thing is here only, and this is the hydrogen bond. We want to break this hydrogen bond. And this breaking of the hydrogen bond can be done with the help of dilute HCl, okay, or some dilute bases or temperature. And in this case, the temperature will be about 92 to 94 degrees Celsius. And we have to keep it for very short time. Because if we will keep it for the longer time, okay, then what will happen? Along with the hydrogen bond, the phosphodiester bond will also break and the structure of the DNA will be destroyed. And that is not our requirement. We have to make the multiple copies of the DNA. For that, we are required to break only hydrogen bond, and that can be done with the help of this much only. Okay. Now, while applying this much of temperature or by adding this one or by adding this one, we have broken down the hydrogen bond. Clear? Right now I am not breaking, so I am going to erase this. And what product we will get now? We will get the two separate pieces of DNA. Okay, how we have got? By applying dilute HCl or the temperature of 92 to 94 degrees Celsius, or by applying some bases. Okay, so this is our separated DNA. What to write? What we will get? We will get separated DNA. Now children, the next point is aniline. Okay, so our second step will be aniline. Clear? Now, the meaning of aniline is hybridization. Okay. Now, if we are talking about the DNA, then what is the hybridization in case of DNA? Okay, what we can hybridize? The meaning of hybridization is intermixing of the two traits. Okay, or intermixing of the two substances. So, what we are going to hybridize? In case of DNA, nothing. It will be only DNA and RNA hybridization. And how we will get the RNA? We will get the RNA in the form of primer. This also I have explained to you while at the time of DNA replication. So, what is this primer? This is a nucleotide of 7 to 8. Okay, primer is a small nucleotide of 7 to 8 pieces of the nucleotides. So near about 7 to 8 nucleotides, they will club together and then they will form the primer. And the primer will first join at these places. If the primer will join, then only the process of the DNA formation will occur. We have already studied this. What is the requirement of the primer? It has the two important functional groups, and because of that, it has a much more space and capacity to combine with the DNA. Clear? So once it will bind at the site of DNA, this I have already explained you. DNA polymerase, DNA primase, epsilon, alpha, all these subunits, they all will help. Someone will help as a primer. They will bind at the site of the DNA. Then there will be the formation of the nucleotide. So the same process will be there. Like normal DNA replication will be there. Okay. So what happens here? But there will be one difference. Clear? So annealing means hybridization, means we have to add the primer. Now children, again the question comes, where to add the primer? Because primer will always grow in its third carbon. In its third carbon. Clear? So because at third place only there is a function group. So the primer will increase from the third carbon. So we have to add the primer at the third carbon of the DNA fragment. And here we will add our primer. This is the primer. And what is it if it is third carbon? Then this will be the 5 prime. And this is the third prime. Because here only the primer can grow. Clear? In the same way. Now, in natural system of DNA replication, what happens, children? There is a formation of Okazaki fragments, lagging strand. They joins the primer multiple times and then the elongation of the chain takes place. But here, because everything is going on in the lab, so we have already broken the whole strand of the DNA. So what we can do, one primer can fixed here also. Okay, if this is third, this will be the 5 prime and this will be the third prime. So the use of primer at first, okay, it should always be at the third carbon of the DNA because it will go from 5 to carbon number 3. Now what are the enzymes which will require? Core enzymes and everything we have studied. Like we will see that RNA nucleotides. RNA nucleotides. Okay, so what is this primer? This is RNA nucleotide and we need the enzyme RNA polymerase. And children, now we need the temperature of 50 to 60 degrees Celsius and almost the approx temperature is 56 degrees Celsius. Clear? So after applying this much of temperature, after applying this enzyme, the primer has been attached at this place. Okay. Now, next step is extension. Only fixation of the primer is not the end of our technique. We have to increase so that we can get the 2 pieces of DNA. Clear? So the third step will be extension. Now I am going to erase this denaturation. Now our third step is extension. Okay. See what happens in case of extension. What things are required? Definitely the chain will extend in case of extension. Now, what is there? In this case, the temperature would be 72 degrees Celsius. Okay. So that the enzyme, and we need some enzymes. Clear? Now, what will be the enzymes? We need enzymes of DNA polymerase. Okay. Now, just see children, at the temperature of 72 degrees Celsius, the desired DNA will not be able to survive. Why? Because our body has a normal temperature of 32 degrees Celsius. And if the temperature increases, then we will not be able to get the desired DNA. So, we need the DNA polymerase of such type of organism which can survive at 72 degrees Celsius. Understood what I am telling you? I am telling you that I need enzyme, and name of the enzyme is DNA polymerase. Okay. Along with this enzyme, we need the temperature of 72 degrees Celsius. Now, from where we will take this DNA polymerase? Definitely, if we will take it from the human's body, then our body cannot survive for 72 degrees Celsius. That is, the DNA polymerase of animals, they cannot survive at 72 degrees Celsius. So, as soon as the DNA polymerase of our body or animal's body has been taken, and if the scientist will use this DNA polymerase for extension, then this DNA polymerase will destroy. Clear? So, we need the DNA polymerase of such organism which can survive at 72 degrees Celsius. And one such DNA polymerase or one such organism is a Thermos Aquaticus. One such organism is a bacteria or a prokaryote named as Thermos Aquaticus. Now, from the body of this prokaryote, the scientist will take out the DNA polymerase. Now, this Thermos Aquaticus can survive at 100 degrees Celsius also. That's why, children, we say that the prokaryotes are found everywhere. You have already studied this prokaryote in class 9th, where you have studied about thermoacidophilus bacteria, which can survive at a higher temperature. So, this is the same bacteria, this is the same prokaryote named as Thermos Aquaticus, and this Thermos Aquaticus can survive at 100 degrees Celsius also. So, what the scientist will do? They will take the DNA polymerase from this organism, and then we will use this DNA polymerase for our extension. Okay. Now, this DNA polymerase is also called as Taq polymerase or Taq enzyme, because we have taken it from the Thermos Aquaticus. So, instead of taking the DNA polymerase either from the human body or from some another animal body, we will take the DNA polymerase for the extension from the Thermos Aquaticus, and we give it a name as Taq enzyme. Clear? So now, for extension we need two things. One is the temperature and second is the polymerase enzyme, and we got both. So now what will happen? This is our fragment of DNA, and this is the primer that I have already added. Okay, now this is 3rd prime, 5 prime, 5 prime and 3rd prime, 3rd 5 and 5 and 3. So extension will be in this manner. For extension, what we want? We want Taq enzyme and we want 72 degrees Celsius of temperature, and both we have got. Okay, now after doing this, there will be the formation of new nucleotides, One will be in this direction and another will be in this direction. Okay. So now we have got our copy and we can repeat this step for various number of times to get the millions of DNA. Understood? One more thing we will require, and that will be, we need magnesium ions for extension. Okay. So this also we have seen in case of DNA replication. We need Mg2 plus for the extension. Now after this we can get the multiple copies. Now what the scientists have got? We have from the one piece of DNA we got, okay, we got the two. Now it can be separated and again they can be made with a new copy. The same process occurs. Okay, once again they all will be separated and again we will get the new copy. So this is the method by which the scientist will increase the number of the desired DNA, and after that what they will do? They will keep it safe for the future test also, and they can continue with the test. Okay, so this is complete steps in case of DNA polymerase, that is polymerase chain reaction. Okay, so till here. Now children, you please go through these steps. Rest we will do in the next class. Thank you.